Introduce Dr. Suzanne Redelinghuys, who's going to be talking to us today about sea urchins and their guts. Sue and I actually go quite far back. I used to demonstrate her practical sessions when she was in second year in 2014, so I've known Sue for a long time. But Sue actually started at SAIAB in 2018, where she was working under Professor Francesca Pori, and completed her PhD under Francesca as well, graduating in 2024. She's completed one postdoc under Francesca and is currently busy with the second one, with research interests varying over the years. So this kind of started from stable isotope analysis in freshwater fish to microplastics in coastal systems, and finally marine rocky shore invertebrates like our friend over here, the sea urchin. So for her PhD, she was looking at sea urchins, their population structure, their gut bacteria, and their morphology, which is all really exciting. I mean, often you walk on the beach and you find your little pumpkins, and you don't really think too much more of it. So it's actually really exciting to see some cool research happening on the sea urchins. And then together, these were used to determine if sea urchins can be used as bioindicators for assessing ecosystem resilience, which is something really important as we move into the future. So this morning, Sue will be presenting on how the gut bacterial communities and sea urchin populations vary across distribution range of the species, which is found across steep thermal gradients created by prevailing currents found along our coastline. So with that, Sue, we hand over to you, and yeah, look forward to the talk.
Good morning, everyone. Thank you so much for taking the time to come listen. I know that you're all very busy. I know it's also compulsory for a lot of you. Thank you nonetheless. So as Taryn said, today I'll be talking about my sea urchin research, which I did for my PhD. And before I start, I want to tell you something about yourself that is not an insult. You are a holobiont. So a holobiont is more than just a single entity. It's looking at the sum of three genomes, which we'll look at in detail in a second. And while the importance of microbial interactions in ecosystems is well understood globally, we don't really fully understand how the internal community can affect the host. So when I talk about internal community, I'm mostly talking about gut bacteria, how it can determine or dictate the host's physiological state, limits, and survival. So let's consider the three genomes now. First is the host genome. So this is the nuclear genome found in every single one of your cells in the nucleus. This is inherited from both parents, paternal and maternal. Compare that with the mitochondrial genome, which is inherited only maternally. And as a consequence, it has far fewer mutations across a similar time span than what you see in the nuclear DNA. Finally is the microbial genome. So this describes the collective DNA of all your gut microbes. So it's not only bacteria, it can also be archaea, protists, etc. The kind of niftiness of the collective gut genome is that the host DNA is relatively fixed. We all know that you as an individual, you can't change your DNA now. A lineage over time will collect mutations or accumulate mutations, but the gut is a very dynamic, or the gut bacteria is a very dynamic community. So that means it can respond to changes in its environment a lot faster than your own DNA can. At its most basic, of course, we know that gut bacteria breaks down ingested material. This brings us back to the holobiont. So by considering all three of these genomes, you are given a very comprehensive account of the biological function of the host, in this case, my sea urchin species. Another introduction is the model or the study species that we've been focusing on, the Cape sea urchin, the Latin name Parachinus angulosus. So this is the story, we like to say, one species, two oceans. It's found across a very broad distribution from, that's not going to work, from Umhlali on the east coast all the way to the border of Namibia. Now, obviously, organisms don't care about political, geographical boundaries, so it actually extends into Namibia and into Angola. It's just that our permit ended at the border of Namibia. What we are expecting to see is a break in the lineage. It has been seen for other invertebrates, specifically off the coast of Cape Town. The physical scale of this species. So it's a medium-sized echinoid. It's very rarely larger than 60 millimeters, so 6 centimeters in diameter. And that's actually quite difficult to determine what age they are. From age 4 years, they invest very heavily into gonadal growth instead of somatic growth. So it's not really a very efficient way of aging them, but we didn't need to age them for this study. Their trophic role, they function as graze feeders. So you'll see in a moment a picture that I have. They are kind of upside down. So their mouth is at the bottom and anus at the top. And so whatever surface they're on, they can constantly graze, constantly gouge at the substrate. Its primary or preferential diet is the mega kelp, the megaflora off the coast of Cape Town. And so it is the preferential diet, but they are, you can describe them as different things. Non-obligate herbivores, detritivores, we can go into any number of combinations of those words. But it doesn't only function as a bioengineer, so changing its environment through its presence, through its graze feeding. It also has commercial applications, or at least commercial benefits. In this case, it is acting as a shelter for juvenile abalone. So we know that abalone off our coast is heavily poached. In this case, it is, shall we say, supporting the biodiversity of the area, because the abalone actually can hide under the spine canopy. They are too large to be eaten themselves, though. But with the sea urchin feeding on detritus and whatever else it can find, what is happening is those bits of food that it is not ingesting is now being ingested by the juvenile abalone. So it's a kind of a twofer. Now, like many species, the imminent most threat for this species is climate change and a resulting increase in water temperature. The steep thermal gradient created, excuse me, by the Benguela and the Agulhas currents, I'll speak about that in a second for those of you unfamiliar, is therefore a perfect, almost a perfect model in itself, just to look at how these urchins can respond to predicted temperatures, because they're found in waters as high as 23 degrees and as low as 8 degrees. So that's a huge thermal range. These are immobile animals, though. They can't simply get up and walk away or swim away. Where they are is kind of where they're stuck. So we need to understand their resilience to changing conditions. And what we are expecting is that the microbes are going to be bringing that resilience, because they can adapt faster, because they have a higher reproductive rate. So this is just to show those of you unfamiliar with the coast. This is a screenshot of what was happening off the coast of South Africa this past Saturday. So I know we have some international listeners and some newly arrived international visitors. On the east coast, you have the warm Agulhas current, and then on the west coast, the cool Benguela current. Another introduction, gut microbiota. I know a couple of you have heard me speak about this at length with the SAIAB symposium. What we need to understand about microbes is, especially in a marine environment, it's found everywhere. It's found in the water column, it's found on the benthos, it's found on every surface of every, not even living thing, rocks, etc., in the marine environment. That means that it can be found on the skin, or it is found on the skin, the outermost layer of whatever species you're looking at, including sea urchins. But that's not what we were interested in. We wanted to look specifically at the gut bacterium. I do want to apologize to the freshwater people in the room. I'm fairly sure that this is a freshwater fish that I've put in a marine environment, but BioRender didn't have many options, so apologies. So this is a really nifty picture to show you what the inside of a sea urchin looks like. It's not my species, but I've tried to get this cross-section. It is very difficult. So what you see at the bottom is the perignathic girdle, which attaches, or ligaments attached to the perignathic girdle, and then the top of this structure, which is the lantern. So it's called the Aristotle's lantern if we refer to it properly. And that's because Aristotle was the first to describe it in what we would consider a scientific sense. From the center of the Aristotle's lantern, it travels upwards to the center of the top of the shell. And that specific section is the esophagus. From there, the gut winds around the inside of the shell all the way around, obviously also on the cross section that is missing. And you can see it looks a little bit gross, but that's because of the preservative fluid that was used. So what you can take away is that there's not really meat inside of a sea urchin. Well, not me, because I'm not interested in trying it, but what the different industries, the food industries like sushi, pizza industries, ice cream industries, they use the gonads specifically. Here it is a white color. Usually it's pale yellow to deep orange, depending on the species and their diet. So looking at what the gut bacteria do, first of all, they digest food, very basic. They optimize nutrient uptake by breaking down things like complex sugars and lignins, and they can even help regulate immunity. This is mostly by outcompeting any negative pathogenic species in the gut. So it's not directly related to immunity, but it does help aid it. Now, this is all of these different components are quite well known for a lot of sea urchin species, but not very well for my Cape sea urchin. Getting into the nitty-gritty, so here, these black triangles represent each of the 45 sites from which I collected sea urchins. We collected 10 from each site, so that gets us to 450 urchins that I dissected, took morphological characteristics for, and again, the thermal gradients that we see here is what makes this such a powerful model. Looking at the materials and methods now, I know that for those of you who are not in genetics, this is quite boring. I'm going to try and make it interesting. We first start with DNA extraction from the sea urchin. So we did that using a modified salting out method. And from there, we then prepared libraries using the EasyRAD protocol. One of my co-authors, Rob Toonan, is the one who designed this protocol, and it absolutely saved my life. For a year and a half, 2 years, I was beating my head against the wall trying to use a protocol that was recommended, but wasn't working. And then EasyRAD came along, and my life was completely different. The next step is purification. The fact that it's magnetic bead purification is neither here nor there. You need to know that it was purified. And then it was sent to New Jersey for sequencing on an Illumina HiSeq platform. By contrast, the 16S rRNA gene is what we were interested in for the gut bacteria. It was then just a basic explanation. It was then amplified using a thermocycler and then sequenced by our fantastic one at Matcha on the Illumina HiSeq platform that we have in the building. There's no difference in quality between the two. The primary, if not only difference, is that the length of the sequences that you get is very different. Looking at the bacteria first, or the bacterial results first, you can see that it's a pretty consistent group of bacteria that we find across all species. There's something weird happening here with Fort Acre, and then similarly with Centilena 1. There were 3 samples from Centilena that we ended up using. So the first one was throwing a bit of, weird results. But before and after analysis, it turns out that there was no effect of these, so we kept them in. What I want you to take away is these black bits at the top represent all the other rarer species. So you can see that we definitely have a predominant or predominantly occurring set of bacterial species. Looking now at the microbial dashboard, so considering first alpha diversity, it was found that there's no significant difference in bacterial richness within each of the coasts, but there is between the 2 coasts. This was a similar finding to the beta diversity. Again, there was a significant community composition between the coasts, but not within the coasts. Looking at our distance decay analysis, so there was a significant correlation between geographical distance and bacterial assemblage. So that means the further apart they are, the more different they are. And then finally, looking at the functional profiles. So this is where you can infer what the different biochemical pathways are that these bacteria are performing. We found that carbohydrates and amino acid metabolism are the 2 most predominant biochemical pathways, which you can see here. So the blue bars represent the west coast and the red bars the southeast coast. And you can see metabolism, metabolism, metabolism. So most of it is metabolism. What we were interested in is this and the phenyl. Oh, I told myself not to say this word because I'll trip over it every time. Phenylalanine, I think. Bear with me. So obviously there are spikes in other areas, but we weren't interested in those for this study. Looking now at how these species occur together, this is a co-occurrence network. It's a really nifty way of seeing which of the species occur together and the relationships between them. So first of all, we have 56 nodes, which are each of these colored circles. So those represent the individual taxa. 154 edges, those are the connections between the taxa, indicated by the arrows at the bottom. And then finally, 18 modules, and those are the interconnected node clusters. So those have connections with others. What we found, is that these four nodes are the ones that occurred together in every single species. So Caritimatus lacey, Bacterodalis, Disulfur bulbaceae, and marine bacterium R52 found in every single specimen that we worked with. Marine bacterium R52, we don't have a name for it. It wasn't properly described, but it was found by other researchers which uploaded it to the database. And so we can at least say that, yes, we found something that is equal, but we don't know what it is. So the conclusion that we're taking away from this is that the diet actually shapes the microbial community in the gut. So that means whatever it's eating, the bacteria already exist on that piece of edible material, which is then taken in and preserved in the gut. Because they are detritivores, it may be that these bacteria are already breaking down these bits of edible material, which is then how the bacteria get in the gut. Because of the distance decay analysis and because we saw the further apart they are, the more different they are, what we also concluded is that while the guts are seeded, the local conditions in the area, so for example, warm water versus cold water, is what's helping shape the microbial community. When I say warm water, cold water, I'm referring to the flora living in those two different temperate zones. Looking at the genome of the host, so the sea urchin itself, the landscape ecological association analysis showed two ancestral populations. So when this graph creates this elbow, it's very strongly saying two populations is what we're working with. Heterozygosity, so that's where you have two different alleles for the same gene. The indices found very little difference between the south coast and the west coast. You can see 0.218 and 0.219. So that means there's probably a lot of mixing of genes between the two coasts. It seems a little bit counterintuitive because the currents are so strong. How can they swim against the current? One of the mysteries. I don't think they're actively swimming. They're carried by eddies and that kind of thing. This conclusion is supported by the fact that there's no isolation by distance, meaning genomic distance didn't correlate with geography. So just because the sea urchins, not the gut bacteria, just because the sea urchins were further apart doesn't mean they were more distantly related. We created this co-phylogeny tree as well to look at the phylogeny based on the genomic, the host genomic analysis versus the microbiomic tree. And you can see that they are very different. So if you were to consider only one in isolation, you'd be getting a very different picture than if you compared the two or included both in your analysis. So we said, found, decided, declared that this is not concordant. We're almost there. The takeaway, decoupling host and microbes. This discordance was the first thing we looked at. So because there's discordance between the host genomic structure and its microbial community, we are saying, again, looking at only one in isolation doesn't give you a full picture. Second, the host population remains genetically homogeneous. Again, that's because of this exchange, this gene flow between the south coast and the west coast. And then the gut microbiota exhibits significant spatial structuring that aligns with specific bioregions. So the bioregions, the coastal bioregions that we considered were those of Lombard, just one author, Lombard 2004, and they identified the Natal bioregion, from the East Coast, Natal bioregion, Agulhas bioregion, Southwestern Cape bioregion, and the Mako bioregion. Analyses showed that there was no difference between the two bioregions of each coast, so we grouped them and just compared coast and coast. What it does point to, though, is that because of the differing temperature profiles, regimes of each system, we were getting different flora, which means that the gut bacteria were different. Please don't use what I'm about to say. I think taxonomists will possibly murder you. You can't say that a host has a generic or general genetic blueprint, but what I'm trying to say is that there's very little difference if you look at the host genome. Contrast this with the microbial community. We have spoken about that at length now. The microbial community is highly sensitive to local environmental filters, so things like water temperature, the type of rock found in the area, the predominant current speed, all of those things are things that can affect the microbial community. And then geographic distance, again, just because they are further related in terms of the host genome, doesn't mean they are more distantly related, but the gut bacteria, it does mean the further apart you are, the more different the community is. So we say that it is a lot more flexible, it's in flux compared to the host's nuclear DNA. Very last slide. So this was the first integrated survey of gut bacterial diversity and host genomic variation in the Cape sea urchin ever. What this research particularly shows is what was happening in the austral summer of 2019. So obviously we would have liked to go back over seasons. That wasn't feasible. But this points to needing to understand, if we are going to understand the sea urchin in its entirety, we need to look at both the host and its microbes, specifically at the genomes that each represent. The microbiome contributes and varies across regions in ways that the genome alone cannot capture. So by losing the microbial component, you're getting a different picture of what's happening if you look only at the genome. And we hope that this is providing a baseline for understanding the hologenome in a changing climate. So I spoke about holobionts, but now also hologenomes. So holobiont is the individual which has each of the three genomes. The hologenome is just looking at the genetic information. The ability of a species to adapt to something like warming oceans, ocean acidification, may then depend less on its own slow evolving genome and much more on the microbial genome, which is, because of a higher reproductive rate, is changing at a much faster rate. So considering the holobionts is important for predicting how marine species will fare under future ocean conditions and ocean stresses. And we hope, or I hope, that through this presentation, I have convinced you that that is important. I'd like to thank the following people, acknowledge the following funders. The most exciting slide of every presentation, the references. And then thank you for listening, you holobionts.
So cool to know that they're like tiny critters living in you and they all do like different things. So amazing. And how they don't get dissolved by stomach acid. Like that to me always blows my mind. Okay, any questions for Sue?
That was a great talk, Sue. I'm wondering about environmental data that would let you go beyond just the kind of regionalization of the kind of west versus south comparisons of the microbiome. I mean, are there actual temperature data, mean annual temperature or something like that, or geological data, or that kind of thing that you can get quantitative on the IBD gradient? I'm
sure that exists. We just didn't record that metadata. This project actually morphed kind of as I was collecting the specimens. So what we ended up collecting, or the information we ended up collecting, is not as comprehensive as we would have liked. So maybe if I get to go back, we'll see. Any questions on that? Okay.
I've got a question. So you showed us that cool cross section of the sea urchin. You said, oh, there's not much meat here. So which part of the sea urchin do you get the genetic material from to do DNA extractions and stuff? Because like fish, you just cut the fins, but then where do you take from the sea urchin?
So you can consider the tube feeds or the esophagus. We looked specifically at the gut because it was a handy way of just doing one DNA extraction, but being able to use the gut itself for the DNA of the host, and then the gut bacteria for obviously the bacterial analysis. So yeah, many different options. I tried them all. Only the gut was actually successful.
Amazing. Love it.
Yeah, thanks, Sue. Interesting for taxonomists like some of us. But my question is sort of a follow-up to what Peter's mentioned, because I'm just wondering whether you collected environmental, you characterize the environmental microbiome as well, because it seems as if you are deducing your conclusions based on what you found in regards to say there are differences between the different coasts. But do you actually, and I think you linked it to say the composition that you found in the gut is linked to what they were feeding on. So I just wanted to check whether you had a chance to also characterize the environmental microbiome to be able to really conclusively make that.
Yeah, it would have been great to be able to take seaweed specimens and I don't know, microbial scrapes of the benthos. Unfortunately, we didn't think to do that at the time. That would have been really helpful. We did take temperature and pH measurements for each location. And we also were going to consider the effect of different pools. So is there maybe very specific differences between the pools in which they are found? Didn't get to do that either. But in the end, so we did use the temperature data that I had, but in the end, we also used the data set that SAIAB has to look at sea surface temperature as well, which obviously doesn't give us as comprehensive a picture because we are interested in what's happening under the surface.
Yes, as a start, like this is an excellent overview. And then if you wanted to answer some in-depth questions, you could almost zone in on like one specific site, answer a couple of questions, seasonal sampling and the environmental sort of biota as well. And so it can definitely be built on, which is really nice.
That's the plan.
Yeah. Who's got money?
Yeah.
Any other questions for Sue?
You mentioned that there were 4 bacterial species that were consistently found in the aquarium. What could be the possible reason for those reports, like consistently?
So we are thinking that those 4 core bacteria are somehow maternally transmitted, and then wherever they end up settling is where the diet brings in these different species, which means that we only have a core bacterial community of 4 species.
You mentioned earlier that there wasn't like a lot of genetic diversity because of the geographic separation, but that the geographic separation led to a lot of microbial diversity. Do you think that could lead to like new species or different species?
Of the host?
Yes.
Over millennia, maybe. What we did find that was interesting, but this is only for the host genome, we didn't find it for the bacterial genome, is that there seems to be, the whole group tends to one clade, except for 4 specimens, which are randomly distributed along our coast, which seems, so there's this separation of these ancestral populations. It's not speciation, so that the species can interbreed depending, regardless of whether it's on the east or west coast. But it points to probably that this is actually a cold water species because the distribution extends so far up the west coast, and that what we were finding were these like stragglers coming down. And it would have been great to actually go into Namibia and go like get some more specimens there and see if there was the separation then between the warm water population versus the whole cold section.
Especially considering the disjunct between so many mobile species between Angola and South Africa because of that Namibian upwelling cell. Not related to this talk, but you were talking about morphometrics. Did you find any differences between some of the samples? So say like right on the West Coast relative to the Wild Coast samples?
Yeah, we did. Not so much in the test. Well, the shell is called the test. What we did, and it took forever, we took all the spines off of every sea urchin carefully, all 450, and then we measured 30 of the largest spines and 30 of the smaller spines. And we found that if you consider only spines, you can actually tell which coast they come from. There's also some evidence to show that the West Coast has larger lanterns, so that's the mouth apparatus, versus the East Coast. And we think that's because of the type of rock that's found on the West Coast. It's a lot harder. So on the East Coast, Southeast Coast, the rock is softer, so it can actually gouge. So it doesn't need a very strong lantern, doesn't need very strong teeth to be able to get edible material from rock.
And maybe kelp versus a softer vegetative, like a macrophyte of some kind. I'm aware it's see tenacity, counting spines. Any final questions or comments for Sue?
With the isolation by distance pattern, you talked about the south versus the west, and it shows an overall IBD with the two domains. When you look at the details, are there specific breakpoints, or is it just a nice smooth pattern, and is it clean, or is it super noisy?
I want to say yes to all your questions. So it was a nice smooth, there was no definitive break to show that there is, depending on this coast or that coast, that you're finding a specific result. So we would have liked to explore that a little bit more. PhD pressure got in the way. But in short, it was a nice smooth change. We didn't see the break we were expecting, the genetic break that we were expecting off the coast of Cape Town.
Yeah, I'm just interested in the link that you referred in terms of maybe projecting systems, our understanding of climate change and all that. So I'm just trying to process the fact that where is the link? How can we arrive at that conclusion? Because you are dealing with a species that is not really seemingly affected by the fact that this one is a colder sort of environment, and the other one is much warmer. Or we are deriving that from the microbial communities.
Absolutely.
Yeah.
From the microbial communities. I can expand, but you answered your own question.
Yeah, so that's what I want to, because you're finding differences in the microbial communities from the different environments. So that's what I'm not framing my question.
So yes, the gut microbiota is what showed the difference between the two coasts. We didn't do any modeling, obviously, otherwise I would have mentioned it. But we are hoping that this information can be used to show that with predicted future temperatures, you can expect to see a change in gut microbiota, even if you don't see a change in the host DNA and the population as a result. I don't know if I'm answering your question.
Potentially use it as a long-term monitoring species. So potentially sampling every year at a certain place and keeping track of the microbiome, then you could kind of track the change in time, assuming a change in surrounding environmental variables, but which would also be to account for those fluctuations in those environmental variables as well. But it's something that could be tracked along with the changes in the gut. Which would be really interesting. I think, lucky if there are no more questions online, we'll cut it there. Thank you very much for taking the time to attend the talk this morning, and join me in thanking Sue for giving a talk.
What? No. Oh, it's a kinesis. Yeah, I would fall. As soon as I but I don't understand. I'll take the domain. I was like, yours. What's it to me? I feel like I didn't.
Sue, thank you very much. Oopsie. Oh, good. Oh, yeah, this is mine.
Yeah.
Good me using this. Where's these?
In how Taryn followed up to my